Hello everyone, welcome to this video. In this one, we will be understanding Rhl Quorum sensing system in Pseudomonas aeruginosa. We'll start with constructing our bacterial cell. You know, Pseudomonas aeruginosa is gram-negative pathogen, and this pathogen is opportunistic pathogen. That means it can cause infections when the immune system is suppressed, right? So in Pseudomonas aeruginosa, to cause infection, there is a system of virulence regulators that are present. In the previous video, we have covered the RHL, not the RHL1 last system. In this one, we will be covering Rhl Quorum sensing system and what happens in that system. What are the virulence factors that are being regulated by this quorum sensing system? The point number one is the last system controls the virulence of Pseudomonas aeruginosa as well as motility phenotypes. Motility means the movement of bacteria from one place to another place, and then it also controls the biofilm maturation. Because biofilm is a step-by-step process. There is a step where biofilm gets matured, and that step is controlled by RHL system. Because it's a cell density dependent quorum sensing system, it's really important for the biofilm maturation of Pseudomonas aeruginosa, right? So we'll start with the RHL system. We'll start with the molecule, which is butyryl homoserine lactone molecule. Before we move ahead, I have something to share with you. This is the platform that we have created. It's called Research Goal. We have various courses, research focused courses that you can complete, and you also get certificate after the completion. You can see all those courses. If you like any of these courses, and you can complete these courses very easily, and you get a lot of resource related to these particular courses when you enroll into these. So I've created multiple courses. Like one course that I've created is masterclass on review papers. You have data analysis related courses. You have courses on molecular docking. You have protein analysis editing courses. So these are some of the courses that I've created, and a lot of my students are getting benefited from all these courses. I'm looking forward to your support for Research Goal, our platform for young researchers like you. So this is the AHL molecule of RHL system. It's a short chain molecule. It has 4 carbon chain attached to it, along with the homoserine lactone moiety, right? So these are the molecules. These are known as BHL molecules, butyryl homoserine lactone molecules. These are released inside, or they are produced inside the bacteria like this. So you'll have many more butyryl homoserine lactone molecules that are produced, and these molecules, they are produced by a protein called rhlI. I means inducer. So this inducer molecule is going to produce a lot of different molecules of BHL, and that BHL is also known as C4HSL. C4 means carbon chain 4 plus homoserine lactone moiety, and it's a short chain AHL molecule. It will have a shorter acyl chain, as well as what will happen that once this molecule is getting released, it can because of its small size, it can diffuse out from the cell like this, right? And depending upon how many cells they are there in the environment, these molecules then can be released, and also they have the ability to enter inside the cell also. So at some time, at one point of the time, what will happen that molecules will be present inside as well as outside, because they can diffuse in and out. And if you have more cells, for example, if you have multiple Pseudomonas aeruginosa cells in that environment, what will happen? You will have more concentration of these AHL molecules, or BHL molecules rather, and because of that, it can activate the signaling pathway. Now, how this butyryl homoserine lactone molecule is being produced? This is our protein called rhlI, which is the inducer for RHL system, right? So that is going to use 2 substrates. One is SAM, which was the same that we have studied in last system. SAM is S-adenosyl methionine. So that is one. Second is butyryl acyl carrier protein. So that's the carrier protein, which is going to be utilized by rhlI, and you will get these molecules constructed on regular basis inside the Pseudomonas aeruginosa cell. So you can understand this is the constructor of BHL molecules, and the substrates, they are SAM and butyryl acyl carrier protein. So acyl carrier protein as well as the SAM is the common substrate for LAS as well as RHL molecules, right? Next is it also catalyzes the synthesis of CHL molecules so that they can diffuse out and diffuse in. continuously. So the activity of this protein is really important because this protein is involved in constructing these HL molecules, and because of that, the concentration of HL molecules it can be higher. Now, what happens is, as this protein is producing these molecules, they can come back and bind to another protein called rhlR protein. So this is the protein right over here. That is our rhlR protein, and these have the ability to bind to a particular site that is present inside the in the RHL molecules. It's called ligand binding site. So something like this, these molecules, BHL molecules, they can bind to this particular site, and this protein will have two different domains. One is ligand binding domain, another one is DNA binding domain. So once this interaction is complete, what can happen? This entire complex can actually move and bind to the DNA inside the cell. Let's take the example of the DNA right over here. So this is the DNA, right? And because now the interaction is stable through the other domain, what it can do, it can bind directly to this DNA. So it will have DNA binding domain also. One is ligand binding domain and another one is DNA binding domain. And because of that, it can stably bind to the promoter region of rhlI regulated virulence factors, right? So this transcription regulator, or rhlR is one of the important target for anti-quorum sensing strategies. So people are trying to find inhibitors against this protein. People are trying to find inhibitor against these proteins, right? So that they can find new antimicrobial strategies that will reduce the virulence expression as well as biofilm formation. Now, virulence expression and biofilm formation, how it will be linked with RHL system. So RHL system right over here is going to utilize this rhlR protein. It will go and bind to the DNA and it will start expression of virulence genes. And those virulence genes are responsible for all the problems associated with the infection. Another thing is we also have efflux pumps. So I do have illustration for efflux pumps. These are the protein molecules that are present on the surface of the bacterial cell. As you can understand with the name, efflux means they are involved in taking the molecules in and out from the cell. So if the concentration goes out of control of these butyryl homoserine lactone molecules, these efflux pumps, they can throw out these molecules outside the cell against the concentration gradient also, right? And these efflux pumps, they are also involved in, you already know that they are involved in antibiotic resistance and all, but their entire role is not that specific. They can be involved in regulating the concentration of various molecules in and out of the cell. So that is really important to understand over here, the role of a efflux pump. Now, next is virulence factors. We need to understand what are the virulence factors. We'll start with the rhamnolipids. So this is the rhamnolipid that is being produced because it's the virulence factor. So once this rhlR is going to bind, it's going to start the expression of rhamnolipids. So rhamnolipids, they are going to get produced. Let me create a few of the rhamnolipids right over here. So more rhamnolipids, they are getting produced. These are going to go outside and cause complication in the host, right? So rhamnolipid, they are getting released. What are rhamnolipid? Rhamnolipids, they are biosurfactants. They are essential for biofilm architecture. They promote surface motility also, as well as microcolony formation. Next is they can also disrupt host cell membrane and they can impair the immune cell function. In addition to that, they can also cause the dissemination of bacteria within the tissue. So you can understand these are the arsenals, these are the virulence factors that are going to control the host system. The next virulence factor that is going to be released when rhlR is going to bind to the promoter region is pyocyanin. So I've also created the schematic for pyocyanin. So let's imagine this is the pyocyanin. So pyocyanin molecule is going to get released and it's going to go out, and more pyocyanins, they will be accumulated in and around the bacteria. And pyocyanin molecules, these molecules, they are also very important virulence factors. The role of pyocyanin is basically to induce oxidative stress in the host cell. It can also impair ciliary function and it can also modulate immune response. And it's known as a redox active phenazine pigment. It's a pigment, and when you grow Pseudomonas aeruginosa, you'll see a blue-green pigment that is being produced by the culture. That's a pyocyanin pigment for you. Next is hydrogen cyanide. So hydrogen cyanide, I've used the small abbreviation for that, HCN. Hydrogen cyanide is also going to get released because that ligand was able to bind to rhlR, and you can see how much virulence factors they are getting released by the bacteria because of that interaction. So hydrogen cyanide is also getting released, and you can see so many virulence factors, they are getting accumulated around the bacteria. And now bacteria is basically really protected from various host factors like immune cells and all, because these factors, they are going to target those host cells. And what is the role of hydrogen cyanide? It inhibits this one. It inhibits the cellular respiration, interferes with the mitochondrial function, and it also contributes to the tissue damage as well as immune invasion. In addition to this, you also have proteases, and proteases are the proteins that are also being released in case of LAS system. But in case of RHL system, you also have control over proteases. Proteases are really common. They are required by most of the pathogenic bacteria. So that's why rhlR system also utilizes the ability of proteases to take over the host system. Proteases, they are going to contribute towards the nutrient acquisition, survival of the host bacteria under the difficult conditions, and as well as the long-term persistence of the bacterial survival, right? So I think we have covered all the important virulence factors, and research is also going on that how a particular tiny cell, which is not that much advanced in terms of genes, in terms of the DNA, is able to communicate with the other bacterial cells and is able to control the expression of all these virulence factors and becomes so dangerous for the host, right? So that is the RHL system for you. I hope I was able to convey all the important contributors of RHL system. I hope you were able to understand how the signaling goes and how signaling basically controls the RHL system. At the same time, I hope you were able to understand the virulence attributes of Pseudomonas aeruginosa RHL system. So mainly motility, biofilm formation, as well as virulence is being controlled by RHL system. RHL system and LAS system and PQS system are the three important systems that are present in Pseudomonas aeruginosa, has been extensively studied in Pseudomonas aeruginosa. There may be other systems. Research is still going on to identify such signaling system, and hopefully in the near future we'll understand more about the social communication system of microbes. All right, that is all for today. We'll meet in the next one.